Yeah, so I guess today's session really arose out of questions I get from people all the time. So it's trying to address some of those common questions. And I guess that's really my role in EnviroDNA is, I guess, chatting to clients and trying to understand what their requirements are and helping to design those surveys. And there's repeat themes that come up all the time. So hopefully I'm going to address some of these today. As a disclaimer, I'm not going to solve all the problems about survey design because it's really impossible, and hopefully you'll understand a bit more about it at the end of this talk. But what I want to do is provide some of the, I guess some of the basics of survey design and in particular anything that's really specific to eDNA. But it's important to remember that even though we're using eDNA and we know it's a very valuable technique, that survey design is critical regardless of what tool we're doing. So today's talk could really just be about, the importance of survey design to achieve your outcomes. So just to give a little bit of background, I'm sure most of you have either had some experience or know a little bit about eDNA, but we know it can be a really valuable tool for a number of reasons, and I guess particularly from a survey design perspective is the fact that it can be really sensitive and also very cost efficient, which means we can do things over big scales. It means that we can do repeat surveys for given budgets and get a lot of replication, which hopefully you'll understand is really critical at the end of this talk. But one of the, I guess, I want to start with probably the most common question I get from people, and that is, how far does DNA travel? And I should say that I'm mostly going to be talking today around aquatic systems because that's probably 90% of what we do. A lot of the stuff we talk about is going to apply to a lot of different systems, but primarily talking about aquatic systems. And we know in aquatic systems, particularly flowing systems, that DNA will travel. And that's a massive advantage. It means that you don't have to be sampling precisely where your target species are to be able to pick them up, because we know DNA is going to travel from downstream. And so it means we've got that sort of spatial reach from our results. So we can pick up things that are a little bit further away from exactly where we're sampling. And we also know that DNA will persist in that water for a little while, and typically it's a matter of days, and that sort of provides that temporal reach. And again, we don't have to be at the precise location and at the precise time where our target species are. So that's some of the really important advantages of eDNA, but it's also important to understand that when you're interpreting your results. So in terms of how far DNA can travel, the short answer is it's really difficult to quantify. But so if we've got our target species in a waterway, this river is flowing towards the bottom of the stream. So essentially our target species is continually shedding DNA. And so we get this sort of, you can imagine it like a cloud of particulate matter. It's going to have higher concentration nearer to your target species. And then as it flows downstream, it's going to degrade, but also disperse. And so it's going to get lower and lower abundance. Now, how far that travels varies a lot depending on a lot of different factors, but from a number of different studies, it's generally going to be between about 100 to a kilometre or so in our sort of lowland rivers. Now, this can be a lot higher in some of our sort of faster flowing alpine streams where the water's a lot cleaner and a lot colder, so your DNA persists for a lot longer. I think there's been studies that have recorded DNA traveling in those systems for sort of up to 10 kilometres, but typically in most of the systems we're working, up to about 1,000 metres is around the limit. So if we're sampling, the closer we sample to our target species, the better chance we've got of picking them up because there's more DNA there. And as we get further and further away, our chance of detecting them is going to decrease until eventually we get to a point where we're not going to detect them anymore. And then, of course, if we're sampling upstream of where they occur, and this can only be, a matter of a few meters upstream, we're not going to detect them because all that DNA is traveling downstream. So you can actually use this to get a bit more precise location as to where your species occur by doing sequential sampling along waterways like this. Now, of course, as with most things, there's a lot of stuff that's going to vary with this. So if we have a lot more flow, that's going to be pushing the DNA further along and also probably diluting it a little bit more. Similarly, if we have higher abundance of species, that's going to produce more DNA, and so it's going to be traveling a bit further. Now, of course, this is a very simplistic model. It's limited by my poor PowerPoint animations. But of course, in a natural system, you're then going to have a variety of different patches of different species and different abundance. So you're going to end up with this mosaic of DNA patches throughout your system. So getting back to what we're talking about today. So why is survey design important for any wildlife survey, really? So one, it's about choosing the appropriate method, and that's going to be based on the species you're looking at and the habitat you're looking at. That might be eDNA, that might be trapping, that might be callback recording, whatever it might be. That's going to be really critical, and it's based on understanding your species ecology and behaviour. But survey design is going to make sure that we get unbiased results. So we don't want to be clustering all our survey locations in sort of the best habitat, because we're going to assume that the species we're looking for is everywhere. So really it's about having scientific rigor behind your results, and it means you get much better data quality out of it, and really sort of getting down to this bottom point, which is meeting your key question unambiguously. And of course, by making sure we get good data, it means that we're making best use of our budgets. And of course, we're all dealing with limited budgets a lot of the time. So really having that strong survey design is just basically going to get a much better outcome for your project. Now, to do that, the first thing we need to do is making sure that we're clear about what question we're asking. Now, we need to make sure that's quite specific. From that, we want to choose, well, what is the metric we're going to use to answer that question? Importantly, what's the spatial extent we're looking at? Is it just at a particular site? Is it catchment-wide? Obviously, that's really critical to designing your survey. And then finally, what's the sort of confidence level you're looking at? Are you trying to detect a particular threatened species because a dam is about to get drained? And so therefore, it's critical that you know whether it's there, otherwise they're going to get wiped out. Following on from that, it's sort of is there a longer term objective for your program? So is this forming a baseline for a longer term monitoring program? If so, what is the sort of level of change that you're going to want to be able to detect? What is your confidence level, your statistical significance that you want to attach to that? And I'm going to give you a few examples of that coming up as well. And then finally, making sure you've got some context. So that might be positive and negative controls. It might be a before and after comparison. In fact, we did a project recently looking at the impacts of the bushfires on platypuses, and before bushfire data, we actually had no power to detect impacts of that fire, even though we surveyed about 700 sites. It was only from a subset of sites where we actually had before fire data that we could show those declines. So really critical to have some sort of comparative baseline to compare against. Again, that might be reference sites, so it might be different types of habitat that you're comparing between. It might be different levels of disturbance, so control versus impact disturbance. So making sure you've got those sort of controls in line in your design so that it gives your data context. And I guess the other thing to think about is that most wildlife is going to be mobile. So, are you picking the right season? And again, understanding that ecology is really important. And this is really relevant for eDNA. So for things like frogs, if you're trying to pick them up using eDNA outside of breeding season, a lot of frogs aren't actually active around waterways. And so you're going to get what we would say false negatives because you're surveying at the wrong time of the year. And that applies to callback surveys as well. There's no point listening for frogs if they're not during breeding season. So again, these sort of concepts apply to whatever method you're using. And when I talk about that question, doesn't need to be overly complex. This is sort of an answer I get all the time. What is it that you're trying to achieve with this project? And it's some variation of, look, we just want to know what's there. And that's fine as a question. But I would then say, well, what is the what that you're trying to look at? Is it a particular species? Is it biodiversity that you're looking at? Because that's going to be really relevant to the types of analysis and the time of year you're looking at. And then following on from that is, let's define what there is. Is that at a specific site? Is it throughout a waterway? Or is it a catchment-wide survey that you're trying to do? This is what we mean about being specific. That's what we need to be. Your question can be quite simple, but we need to be able to define it a little bit better, and that's going to inform your sampling design. So when we're talking about survey design, there's really two, things that are relevant. And first of all, it is your sensitivity. So what is the probability of whatever method you're using to be able to detect your species or to quantify your biodiversity? If your method isn't very sensitive, you need to do repeat surveys to be able to achieve a given confidence level. And I guess that's where eDNA tends to really shine through. We know from a number of studies where we've compared eDNA with traditional survey methods that to detect species, eDNA tends to be much more sensitive. So yes, I guess that comes down to choosing the appropriate method. And as I mentioned before, this is going to be based on some knowledge of your species ecology and behaviour. And then, picking an appropriate season, either because of their behaviour or migration patterns or whatever that might be. And typically, even if species are there all year round, there's going to be different times of the year where they're more findable, I guess. They're more easy to detect. For most species, that's going to be around breeding time because they're a lot more active. They're moving around a lot more, and therefore, regardless of what method you're using, they tend to be more detectable. Something that's very eDNA specific is the type of analysis you're using, and the two main ones are using your qPCR, which is a single species approach, or your metabarcoding, which is for biodiversity assessments. Now, there's different levels of sensitivity between the two, so there's pros and cons of using either of those techniques, and that's an important decision to make very early on. Something that's really critical, regardless of what method you're using, is replication. So if you visit a site once, whether it's for trapping, whether it's for observation, whether it's using eDNA, what is your chance of detecting whatever you're looking for with that single visit? Now, with eDNA, we can take multiple samples with that visit, and that's going to improve your chance of detecting that species. And I'm going to show you some examples in a minute. And then, of course, your species abundance. If you've got something that's a really low abundance, of course, you're going to need more survey effort to achieve that confidence of whether you can detect it or not. And also that behaviour, are they quite cryptic? Are they quite static? They don't move around much. That's going to all influence your sensitivity of your technique to detect that species. So this is possibly the second most common question I get is, well, how many samples do I need to take at a site to detect my species of interest? And even though there's a lot of things that influence that, and I've just kind of listed a few of those things here, I'm going to show you some actual good data that shows it's remarkably similar between species. But obviously things like waterway size, a small creek versus a giant river is providing very different environments. And so, if you've got a lot more water, you're going to have to tend to take more samples to achieve that confidence. Understanding a bit about your species, again, coming back to their ecology. So we find particularly things like reptiles tend to shed less DNA. So there's going to be less DNA in the water to find, therefore more replicates are going to be required. Or things like terrestrial species, they're not around the waterway as much, and therefore they don't leave as much DNA behind in the water. So therefore you might sort of bump up your replicates there. Abundance, as I mentioned before, if they're a very rare species, then you're going to have to put in more survey effort versus those highly abundant ones. What's the level of confidence that you require, as I mentioned earlier? And then something that often you can't really factor in the early days is those filtration volumes. So regardless of how many replicates you're taking, if each replicate is 50 mils versus a litre, your chance of detecting that species in each sample is going to increase the more water you're able to filter. And unfortunately, there's not a lot that we can do about that filtration volume apart from just simply taking more replicates. So let's get to some actual test cases. And for those of you that have heard me talk before, you'll know that this is my sort of specialty species, the platypus. So there's going to be lots of examples of platypus throughout this talk. But in the very early days, when we were looking at how useful is eDNA going to be to detect platypuses, we did some fairly significant investigations and some modelling to sort of understand how useful it's going to be. And what we found is that we can go and take just 2 water samples here on the left. And from each of those water samples, we tend to run multiple assays on it. And so we can do 2 or 3 assays is what we tend to use as standard. So from our 2 water samples and just 2 assays, we get over this magical 95% detection rate. So if the species is there, we've got a very high probability of detecting it. Obviously, that's going to vary a little bit depending on their abundance. But the flip side of that is that if we get a negative result, we've got high confidence that the species is absent. Keeping in mind that that's saying that the species is absent at that site at that particular time. Now, platypus in particular are highly mobile, so we could go back a week later and actually find them there. And that's where your broader survey design becomes important. So we've done this for a number of different species. We found very similar results for cane toads. So just a couple of samples for high detectability for cane toads. A threatened native fish, the dwarf galaxids. We were looking at probably 3 samples per site to get over that 95% detection. And for Macquarie perch, again, we're at that sort of 2 samples per site. So remarkably similar across a range of different species with different behaviours. But again, it's going to come down to your particular species of interest and the environment that you're looking at. But generally, as a baseline, we tend to recommend 2 to 3 samples per site as a starting point. Similarly, if we're looking not just at target species, but we're trying to understand biodiversity at an area. So this was looking at fish biodiversity. Each of these little graphs is a different site. And we've got the number of replicates here and, creating these species accumulation curves. And again, 2 to 3 samples per site, we're detecting most, if not all, of the species that are present there at that particular time. And again, this is important to remember that, species are going to move around, and even though we're detecting the species that are there at this particular time, at a different time of the year, there might be different species that come on board. So, spring and autumn in combination is probably going to get you a much or a slightly higher estimate of biodiversity than just looking at a single point in time. So that's probably a good point to have a quick break and see if there's any burning questions that we can address before we launch into the next little part.
Thanks, Josh. We have had a few questions come in. We probably won't answer them all now. We'll just pick out maybe 2 or 3. So we've got a question here that says, how do your expectations for species presence affect your sampling methods?
Sorry, can you just repeat that one, Jackie?
It says, how do your expectations for species presence affect your sampling methods?
Okay. So I guess if your expectations are that your species are going to be at high abundance, then you've got very high probability of picking it up, probably with most techniques that you're going to use. If they're at low abundance, that's when it's really important that you've got repeat surveys, spatial replication, targeting, understanding a little bit about the ecology and targeting the particular habitat preferences for that species. but really it comes down to what is the key question that you're trying to ask. You know, is it just, is this species present within this particular area? Are you trying to have some sort of measure of a population metric? So, you know, we tend to use site occupancy for platypuses, which is a sort of surrogate for abundance. So hopefully that helps.
We've had a couple of questions come in about replicates and endangered species. So how many qPCR replicates is needed to consider a positive eDNA detection for an endangered species?
Yeah, good question. So for each sample, we tend to do 3 technical replicates in the lab. So if we've got 2 samples per site, we end up with 6 assays for that particular site. It really comes down to what level of confidence you require. So as a result from those, and Luke's going to talk a little bit more about this next week, but so you can get anything from 1 to 6 positive assays from that site. The more positive assays you get, the more confident you are that that species is present. We tend to set a threshold of at least 2, which some people would consider conservative, some people would say it's not. We know that you can get very low levels. So, you know, you can sometimes get very low positive results that aren't real. That can come from a variety of sources. Most commonly, that's going to be contamination, either in-field contamination or in-lab contamination. Now, we set a lot of strict protocols to minimize that and to detect it if it's occurring. And then also you can get, depending on how well your assay is designed and your probe is designed, you can get cross-amplification with other species. Now, this tends to happen at very low levels, and that's why we set those thresholds. So yeah, we would tend to go at least 2 out of the 6 or even 3 out of 6, depending on what level of confidence you require. It's also understanding a little bit about what other species are present and, you know, how specific your assay is. So I guess that's where, you know, something that's very specific to eDNA and making sure you've got that level of verification that your assay has been tested against a variety of different species. The more closely related species are, the more difficult it is to tell them apart from a single assay. But yeah, we can certainly, you know, talk through that and help with that.
We'll just do one more question for this Q&A, and we might leave the rest until the end. So do you have any data on the movement/dispersion of eDNA in lentic systems?
Not a lot. There are a few papers out there. I mean, logically, it's going to be, tend to be if you've got flowing water, it disperses. It tends to be fairly homogeneous throughout the water column, and there's several papers that have looked either across a river or depth-wise and found really limited difference. In lentic systems, so in you know, wetlands, lakes, dams, things like that, logically it's not going to move around as much. We tend to use a slightly different sampling approach there. So you can either, you know, make sure you spread your sampling out throughout that system to try to cover off on that, target your habitat for your target species as well. But it's going to be a lot more clumped and clustered within those systems. So, and I guess that's just about understanding how DNA moves and, you know, the differences between your systems and a bit of the hydrology as well. So, but yeah, certainly using a slightly different sampling approach for lentic systems is worthwhile.
Yeah. Okay, cool. We've had a few other questions come in, but we might leave that there for now. And Josh, I'll let you continue. Okay.
No worries. We'll get to those questions at the end, hopefully. And there were a few I saw that looked technical. I would say I'll do my best to answer them, but I think if you want more technical answers, maybe listen in to Luke next week. That's more his specialty than mine. But okay, let's move on. So getting back to our survey design. So once you're understanding a little bit about the sensitivity of your methodology that you're using, then I guess the other component that needs to go into your survey design is your power. And that might be, what's the power to detect your species? So this is a little bit different from your sensitivity. So sensitivity is the probability of detection at the site level. Your power is more about the probability of detection across your survey area. So that's really related to site replication and then also site repetition. So as I mentioned before, wildlife's going to move around just because it doesn't occur at a particular site at this particular time. It might be at another time. And so yeah, understanding a bit about that, and whether you want to do spatial replication or temporal replication is going to give you slightly different outcomes. So again, we're back to my river animations using platypus again. So if my question is, and I'll go back to that simple question I asked before, is we want to understand what is there. In this instance, is we want to understand whether platypus occur within this river system or river reach. Now, if I go out and take a single sample at a single point, yes, there's a chance that I might detect them, but I'm most likely not going to from a single sampling point. And we all know n=1 is to be avoided at all costs. If I then, sample at multiple points along this system, the more sampling events I do, chances are I'm going to detect them somewhere along that system. So even though, this is more talking about do platypus occur within this river system rather than do they occur at a particular point. So having that site replication is going to be really important. But then also when we think about different species abundance, I would say this scenario is a fairly low abundance population. If we have a high abundance population, your chance of detecting them is much greater. And so, my single point is most likely going to pick up platypuses. And then if we look at multiple points, most of them are going to pick them up in a high abundance area. And this is where we can start using metrics like site occupancy to try to understand our population health or our population abundance. So while we're not estimating absolute numbers for something like a platypus that's very widespread and tends to be at low abundance, something like site occupancy over a larger area is probably a more relevant metric. And we can see how that changes over time to work out whether populations are increasing or decreasing. Similarly, if we want to be looking at our biodiversity, so we want to be able to detect as many species within our river system or our subcatchment as we want to. In this instance, it's looking at a vertebrate assay. Sorry, I've thrown in a few invertebrates there, but it was really just to show a range of biodiversity. So this was some modelling we did recently along a river system. And we found once we got beyond 4 sites, randomly selecting 4 sites from within this river system, that was enough to detect all of the species that we were going to within that system. So again, using sort of some of that data to understand, creating species accumulation curves to understand what's the minimum survey effort we need to be able to do to achieve our goals. Now, some of you have probably seen this map before, but a while ago we did some fairly widespread surveys for Melbourne water. And so when we talk about the power of our survey design, one of the things that we're often trying to determine is, can we detect change over time? So for a lot of our long-term monitoring programs, that's really what we're trying to do, whether that's trying to understand that change due to natural variation, whether it's a change due to disturbance events like a bushfire or a flood, or it might be trying to understand how effective our management processes are, and are we seeing an increase in our target species from that. Regardless of what it is, we're trying to detect change over time. So a while ago, and this was back when we were trying to really develop eDNA as a technique, we did these widespread surveys mapping platypus distribution across the Melbourne area. So across 5 river catchments, it was about 330 sites, I think. Orange is where we found platypuses and white is where we didn't. There's some pretty clear patterns there, and you can kind of see how useful this would be to look at sort of habitat types and, you know, all kinds of spatial layers you can look over. And there were two goals to this project. Firstly, it was to produce this map, and so that Melbourne Water can understand at their sort of broad scale where platypuses occur and where they don't, so then they can target their management actions. But following on from this, their longer term goal was for this to be a baseline to then detect change over time. So we were able to use this preliminary data using some site occupancy models and power analysis to go, okay, well, if we wanted to detect a given change between time point 1 and time point 2 with a certain level of confidence, how many sites do we need to survey? And so, you know, we can produce these sort of useful things where looking at different levels of change, so 20, 30 or 50%, and whatever statistical power we want to look at, we can very clearly go, well, these are the number of sites that are required. And obviously the more confidence that you require and the greater or the lower the level of change is going to require more sites. But so for that current survey level, we're at here at about 330. So we can pretty much detect a 20% change with 70% confidence. But if we want to get up to that magical 95%, we're going to need a lot more survey locations. And Melbourne Water have now taken this on board for not just platypus, but for a variety of species, and have implemented a very intensive eDNA program across these catchments based on this sort of scientific rigor that we've put behind that. So, you know, they're very confident that their investment is going to produce a usable outcome for them. So I guess just to recap, you know, survey design is critical to answer your question. Now, this doesn't mean that we necessarily have to be able to quantify a lot of those things, but understanding some of the stuff that goes behind that in terms of your sensitivity and your species ecology is going to be really critical to design something that's going to give you usable data. I guess the most critical thing, and I can't stress this enough, but you need to be able to define exactly what it is you're looking at. And as I said, it doesn't have to be a complicated question, but you do need to at least decide specifically what it is, what's your spatial scale and your temporal scale, and then try to understand, well, what are the metrics that you need to achieve that? That's going to really guide what your methodology is going to be, as well as your sampling design. Sitting behind that is really understanding your species ecology. So it becomes a lot more complicated when you're doing biodiversity assessments. It's really difficult to target something that's going to be relevant for all your species. But certainly for when you're looking at single species or a couple of species, understanding things like their ecology, their behavior, you know, when are they going to be most active at different times of the year? What are their sort of habitat preferences? And that's going to inform your sampling design. Thinking carefully around timing and season. So, you know, doing repeat surveys over time is really important, but, you know, often you don't have scope to do that. So can you get around that by doing spatial replication within your area of interest to understand, well, you know, are they there or not? If quantifying some of that stuff, like I've presented, is important, then really a pilot study is going to be critical. And if you're designing a long-term monitoring program, a pilot study will often save you money in the long term, because you're going to understand, well, what is the minimum survey effort that's required, and so that you're not actually, you know, wasting money doing extra surveys that, you know, simply not going to give you any additional data. And from all that, you can then sort of get into the nitty-gritty of your sampling sites. So where and when are you sampling? How many survey sites are you doing? What's the sort of replication that you want within a site? I can't remember where I was going there, but I guess the key thing, you know, with any survey design is really replication. There's a number of reasons why replication at site levels are important. So sample replication within a site is important, as well as your site replication over a spatial scale is going to be critical. All right, I think that's about it from me. As I said, you know, the goal today wasn't really to give you all the answers, but hopefully it's to give you a little bit more understanding around why survey design is important, and a little bit of knowledge to then apply for your specific situations. But I guess the key thing is, if you've got a question, have a chat to us. That's what we're here for, to try to understand exactly what it is you're looking for, and then try to design something that the best outcome that we can, that is then going to be able to get shoehorned into a budget or a logistics to really get the best data we can out of it. So thanks very much, and we'll answer a few more questions.
Back to some questions. So I've got roughly 5 minutes. So I like this one. It was, can you tell whether you are simply discovering the same animal over and over again?
So you can't identify individuals from eDNA surveys yet. I think the field is developing incredibly quickly, and there were some really interesting talks at a recent conference where people are starting to be able to do that, probably well beyond the scope of most project budgets at the moment. So I guess that comes back to sampling design and understanding your ecology. So if we think that eDNA is only going to travel for about 1 kilometre, your species doesn't move around much, then you might set 1 kilometre between your sites as the minimum distance to consider those sites independent. So that means that you're not detecting those same species. For something like platypus, I know they're incredibly mobile. I know they will move multiple kilometres within a night. So I would spread that out. Most of the times I tend to have a minimum of 5 kilometres between sites, because it enables me to, one, ensure that independence between sites, but also spread my sampling sites out a lot further and get data over a much bigger spatial scale. But yeah, that is critical, and certainly having independence of sites is important for a lot of the statistical analysis that you might want to do.
So another one here is, how many samples do you need to confidently assume a species is absent from a site?
Depends on what you mean by confident. So, those graphs I showed you before around, basically 2 to 3 samples to have a 95% chance of saying the species is there. You can flip that around and saying, well, if we get a negative result, then we are 95% confident that the species isn't there. If you want 99% confident, then that sample number is going to go up dramatically. If you want 100% confidence, then you're probably going to have to drain the system and look in detail. Trying to prove something is absent is incredibly difficult to have 100% confidence. So it really comes back to the sensitivity of your technique. So, for example, I know I can go out to a river and look for a platypus. My chance of detecting them is really low. So I would have very low confidence. If I don't see one there, I've got very low confidence that they're not there. Whereas with eDNA, I've got incredibly high confidence of detecting them. So therefore, I have high confidence of saying they're absent if they're not there. But again, I know that they're mobile. So I would say at a site level, that's not really important. If I get negative results over a number of sites along a waterway, then I've got good confidence that they are absent from that waterway.
Yep. We've had this question a couple of times, so it's probably a good one to go back to. It's just, what's the difference between qPCR and metabarcoding?
Okay. So qPCR is a technique that's used to detect a single species. So we develop a genetic probe that is specific to whatever your target species are. And if it's been verified adequately, you've got high confidence that it's not detecting anything else. Whereas metabarcoding is trying to detect entire taxonomic groups. So we can use general probes to target, say, all fish, or all vertebrates, or all decapods, whatever that might be. It's a very different technique in the lab. So we're actually sequencing data rather than just throwing a probe in there and seeing whether it attaches or not. So it gives you very different outputs, but obviously amazing data for a range of different species. We know that it does lower sensitivity a little bit and tends to be around that sort of 20 or 30% reduction where we've done comparisons. And that's simply because you're getting data for a lot of different species. So sometimes your low abundance ones can simply get dropped out. So yeah, very different approaches. I guess it's similar to if I'm doing a bird survey and I do a callback for a single species, I'm only detecting that species. Whereas if I walk through a forest and try to visually record everything I see, that's the kind of difference between qPCR and metabarcoding in a very bad comparison, I think.
So we've got one on platypus. How does eDNA compare to previous systems of trapping platypus? Have you done both together?
Yeah, absolutely. There's actually a paper that reports on it, and we looked at both sensitivity, cost to detect change over time, and eDNA is basically orders of magnitude more sensitive and more cost effective. Produces very different data though. So, for example, I can't tell you sex, I can't identify individuals, I can't get any information on, say, litter entanglement, which is something that, you know, people like Melbourne Water are very, want to understand. So it comes back to what is your question? If you want to detect changes in a population over time, then if it's over a large spatial scale, then you're going to want to use eDNA. If you're trying to look at changes in abundance at a very localized area, then things like site occupancy aren't going to be relevant because you're not going to be able to fit enough sites in. So you might want to do trapping over time. So yeah, again, I can't stress it enough. Define what your question is to start with and then pick your appropriate method.
We have time for one more question. So this one sounds interesting. It's, do you find that certain water quality parameters affects rate of degradation of DNA? And how does this affect your survey design?
Yes, it does. There's lots of things that influence DNA degradation. So things like, UV is one of them, temperature is one of them. We know some bacterial activity is important. Acidity is another one. We don't tend to do direct comparisons there, simply because there's a lot of other things that come into it as well. But yeah, I guess if you need to understand that, then you can look at that. But I think there's enough in the literature to sort of say, well, look, we know these things are going to increase degradation, but really it's a matter of DNA is going to persist for 1 or 2 days versus 5 or 6, and is that relevant for your question? Yeah, so it's not going to get to the point where you're not detecting the species. It might reduce the dispersal distance that you're going to pick them up over. But again, if you've got a strong survey design, it's going to be able to cope with that variability.
Yeah. We might have to leave it there because we're a minute over. But thank you for all of your questions, everyone. And if we didn't answer them in this session, we'll get back to you afterwards individually and we can hopefully provide you with a response.
Yeah, can we save all the questions, Jackie, so that we can then respond afterwards?
Yeah, we'll save all of them and we can get back to you individually.
Okay.
So we'll sign off here. And just as a reminder, we have the webinar coming up next week, which is how to interpret results and get the most out of your eDNA data. And that will be with our molecular lead, Dr. Luke Noble, at 12 p.m. So if you want more information on that webinar and registration, you can head to our website and we hope to see you there. And thank you, Josh, for leading that session.
Thanks everyone for attending, and we'll try to get back to you with your questions.
Yeah. Thank you, everyone.